This video is on a modern method for DNA sequencing. It relies on PCR and electrophoresis. So those are covered in the previous two videos on PCR and DNA profiling. So DNA sequencing obviously allows mapping of species genomes. So we're able to sequence large amounts of DNA, and the technology and the price has come down, and the technology has improved enormously since the Human Genome Project. So some of the details are here, but I'm going to go through this picture mostly and have a look at it. So just a reminder back to PCR, the normal way in which that works is we have a DNA sample, primers, free nucleotides, and a polymerase. And as we go through those thermal cycles, the polymerase adds nucleotides using the DNA template and makes copies and copies of the DNA. The sequencing version of PCR is slightly different. So we've got 4 copies of the PCR going on. So instead of just one, we've got 4 different tubes. Those tubes have what's in here, plus 1 dideoxynucleotide. And so this one has dideoxy-T, this one has dideoxy-A, dideoxy-G, and dideoxy-C. Each one of those is given a fluorescent label on it of a different color. Red, blue, yellow, and green here. So what happens when the PCR starts and we go through the cycles? Well, this is our template of DNA and we're copying this. And under the normal circumstances, the polymerase would keep just copying that and get the whole lot copied. And it's adding free nucleotides as it goes. But what we have here is a case where the dideoxy-G is what's called chain terminating. It stops the elongation of the DNA polymerase. DNA polymerase adds the G, and then it can't add anything beyond that. So the concentration of the dideoxy-G is quite low in that one, so it does have other G's in there, normal G's, and it adds those. But sometimes, occasionally, and on this one, what it's done is it's gone along, and the second G it's hit, it's put in the dideoxy, and so it stopped there. On this fragment, it went along, and the first G it went to add in was the dideoxy, and so it stopped there and didn't add any more. So what's happening in this reaction mix with the dideoxy-G is that randomly, as it's going through, it makes long ones, short ones in between, but sometimes whenever it hits a G, it stops. And so we end up with lots of different sizes of DNA in here. Each of them ends in a G. So that size corresponds to where a G would go. Same sort of things happening with the A here. Randomly, it'll put in a chain terminating blue fluorescent A at wherever an A position should be. And so once we get these 4 different reaction mixes and we run them through electrophoresis, remember that electrophoresis, you put in a mixed sample of different sizes and it'll separate them out based on size. So there are a very small piece down here and some larger pieces up here that all ended in a T. So they end up in this T lane that's red fluorescent with a T position here and a T position here. 3 different positions where the A could have been, and they get shown here. So each band represents a length of DNA that would terminate where that dideoxynucleotide is. You put all those together, shortest one's the red one there, and then C, G, A, so that's the T, C, G, A we see across there, and you get this pattern of bands with the different colors, and this is the electropherogram version. So this is what you end up with, is when you look at all the bands in their position with different lengths, you see each color represents the different nucleotide that would be in that position that corresponds to the piece of that length. And then the piece that's slightly longer with a different nucleotide, the piece that's slightly longer with a different nucleotide. So when you have the pieces all lined up by size and you know which nucleotide they ended with, that's the way you work out the sequence. Have a look at this video if you're not sure. I know it's complicated understanding, maybe go through a couple of times, but this video should also help you understand the chain terminating. But basically what you're saying is you end your PCR at different nucleotides, you know which nucleotide you ended on and how long it is because of electrophoresis. Put that information together, you get the sequence.